Honestly, you can hear everything. So the audience is talking, you can play, man. No.
I just stopped now. With the mic, is it off center?
I'll check.
That's why he has to play. Yes. Meet Dr. Vaishali. She is from RV University.
Hello, madam.
Good morning, everyone. It's a pleasure to welcome you all to the ISGCT AIML 2024 conference. It's the sixth edition of ISGCT and second edition of AIML Bio. Today's track discussion is on one of the most cutting-edge intersections of science and technology, artificial intelligence and machine learning in the field of biology. In recent years, AI and ML have revolutionized many aspects of biological research, from genomics and drug discovery to precision medicine and beyond. Today, we are fortunate enough to have among us some remarkable minds who will be sharing their insights and expertise on this fascinating subject. I now request our distinguished panelists to kindly come upon the dais. I now request Dr. Vidya Niranjan Ma'am, HOD, Biotechnology, RBCE, to kindly address today's gathering.
Thank you, my dear students. Genesis. This particular conference is an international conference. This last year we have done AML Bio, so that is the first edition. This time we are going to do the second edition. You all know that it needs a huge funding to do any of the conference. So initially the budget we did is 29 lakhs, and we thought that before starting we should have the funds, which is very important for the conference. I really have to thank Dr. Agrawal Eye Foundation. Gene Research Foundation is one of their NGOs where they are doing research. They told that we want to collaborate with you for a gene therapy conference. So immediately I thought they are ready for funding for their conference and they want collaboration with RV. Then I thought this is the best platform I can get where we are into bioinformatics and they are in the application of gene therapy. So I said, why don't we together do this? So they have happily agreed, and I have to really thank Dr. Sunita Rana Agrawal, who is the CEO of Gene Research Foundation and also a part of Dr. Agrawal Eye Foundation, which is a huge, as you all know, that is a very big hospitals, eye hospitals in globally, and also the Pankaj, who is the CEO, who both told that, and I think this cannot start without thanking them. And we had a very clear bifurcation that they will be completely funding this. The scientific part I will take up. So yesterday was the day one pre-conference we had, where we had the poster presentation, and we had a good number of people from ISC and other colleges like St. Joseph's Christ, our sister concerns, and it was wonderful yesterday. And today is the day two of the pre-conference, so where we are going to have the panel discussion. And I have to apologize to all my panelists. Initially, this was decided to keep it in the main conference only, but there are a lot of speakers and we need to extend the time. And this panel discussion is very important scientifically, so we thought that we will take it as a pre-conference before. And thanks all the panelists for understanding this and accommodating us. And so we took this as a off from the main conference as pre-conference. And today now the theme is on AIML Bio. So this was our second edition of international conference. And I thank all of you. And we are expecting already 600 registrations, students registration has been done. And we are looking out at least 1,000 people to come to the conference. I think a single body of taking 1,000 students into the conference is phenomenal. And thanks for Agrawal Eye Foundation, where they told that they can completely fund the food and not the stay, only the food for the students and to attend the conference. So we have, it's free, but only we take as a registration as 100 for some expense where we need to give certificate. And if students do not need certificate and they want to attend the conference, they are welcome for free also. So this is what this Dr. Agrawal Eye Foundation has given, and we are all working very meticulously. And I think this conference, the funding is a major chunk, and the second is the work force. And I think the Genesis, and especially the 6 core members, I think the relentless work, professional way of hand, because as you know, there are 2 big entities, RBCE and Agrawal Foundation. The culture itself is different, and bridging both together to have this happen, I think thank you is not the word. It's much beyond, and I think I'll stop. I just want to tell, since it has been online, I want to tell what is the things, who are all the players back, who has worked on it. Definitely the funding agency, and the funding agency also has given funds for us to go to Delhi, to other states, to invite people, speakers, and also it also included that. So I think they are doing more than what was given as a budget for them. And also we look forward for a very successful conference, 20th and 21st. Please, all of you, your friends on biotechnology or any domain can come and join for free. They don't need any certificate, but if they want certificate, they have to register the minimal of 100 rupees for the corporate expenses that we are doing it for other things. So I think I'll not hold more than this, and I'll give it to my Genesis team, who is the people who are doing this conference. Thank you.
Thank you, ma'am. Now, before we begin the session, I call Anirudh RRS, the student of third year biotechnology and the head of Genesis, the student-led biotechnology group of RBCE, to moderate today's session.
Good morning, respected panelists, and good morning, everybody. So we have with us esteemed panelists for AIML in Biotechnology Track 1. So we have 3 of them who have joined offline, and as you can see on the screen, we have 2 of them who have also joined online with us. So we'll start directly without any further ado. So I would like... Yes, I would like to request all the panelists to one by one just introduce yourself, especially with respect to the work that you're doing and the main topic of interest that you work on.
Good morning, everybody. I am Vaishali. I am from RT University School of Design and Engineering. So, my area of interest is basically application of machine learning and artificial intelligence in various domains of science. So, biotechnology, both physics, bio, and other related topics. I will because there are a lot of areas where AI have not gotten exposed much. So that is the area of research in the language.
Good morning. I am Dr. Prashant, Associate Professor at Department of Biotechnology, Dynamics College of Engineering. So I work mostly on drug designing aspects, like virtual screening or identifying lead molecules for a target. So I have multiple collaborations, majorly working on now related to gut microbiome to activate some key receptors so that the obesity or any other related diseases can be under control. So, like AIML, a part which will be applied in the whole process, right? And more into drug design aspects. Thank you.
Hello, everybody. I am Dr. Shanta Rangaswamy from Department of CNC RICE. I have been working in this organization for past 16 years. My research interests are AIML, image processing. Basically, I usually work on satellite images. When we say image processing, there are a lot of images. Mostly concentrating more on satellite images, apart from healthcare domain also. Right? So my AIML I have been applying for the healthcare domain, specifically for the neurological diseases, right? Alzheimer and epilepsy, those kind of things, and as well as for the traffic management. Right? Reinforcement learning, I have been applying for the traffic management scenarios. Right? So this is a very good platform where we have computer science and biotechnology coming together, and where it's a must, right? Individually, neither biotechnology nor computer science can take the credit. Only when they put hand in hand, it goes together. It's good for both of them, right? It is like Anna Sambar. Only Anna will not help, or only Sambar is going to help us. Both are definitely required for a delicious food. And that is why technical and biotechnology department need to definitely come together. It is a win-win situation for both of us, right? With this, just a brief introduction, maybe we can start with the panel.
Thank you so much. I request BM Sagar sir to unmute and introduce. So just 1 second, you're not audible.
Hello, am I audible now?
Yes, sir.
Okay. I am Professor B.L. Sagar, heading Information Science and Engineering at RV College of Engineering. This is my 20th year at RV College. Currently, I am heading the department. My research area includes natural language processing, basically the language processing techniques that we adopt here, translating from one language to another language. And we also work on data analytics. And the department is also working on a lot of consultancy projects. And we also have coding club in the department, and students are also more enthusiastic to work towards such projects. And as Madam told, this is the perfect blend of computer science and then biotechnology. Since AIML are AIML algorithms which are so popular now, which can predict a lot of things, so this will certainly help lot of domain, so including biotechnology. And biotechnology is in forefront now to get the benefit from the AML algorithms, and then data intense projects that if you are thinking, I think AI and then ML algorithms are really very accurate and apt for to predict all such things. Thank you very much.
Thank you so much, sir. Now I request Ramakant sir to unmute, and if you can turn on your video, it will be really nice, sir.
Very good morning to all of you. This is Dr. Ramakant, heading Computer Science Engineering Department, working since 1994. So next month, October 7, I will be completing 30 years at RVCE. And I've been associated with many projects in the DRDO organizations as well as private organizations. And my area of research is computer vision, pattern recognition, image processing, and also including AIML algorithms. So I worked on the optical character recognition, ELDA language, and also NLP, natural language processing. When you do OCR, we get a text. It may not be 100% accurate. So by applying the NLP methods, how to improve the accuracy of the recognized things. So apart from that, we worked on the Naval Research Board projects, underwater image processing, and also for VSST, the Return Survey Space Center, one project we have done it on the robotics. Apart from that, we did projects for Center for Airborne Systems and also for AICT, etc. And under me, I have 14 scholars have completed their PhD work in the areas of NLP, the areas of artificial intelligence, machine learning, etc. So, thank you for giving me an opportunity to be the panelist. Over to you. Thank you.
Thank you, sir. So before we begin this panel discussion, I would just like to inform all of you that this entire session is being live streamed on YouTube, and we have almost 600 plus participants who are going to be witnessing this entire event. Okay, so now let's start off with the panel discussion. And this panel discussion is actually going to be really very unique, because usually in all panel discussions, if you have noticed, the moderator just frames the questions and it's posed to the panelists. But here we ensure that we directly get our student coordinators to collect questions that students have in their mind. And it's our pleasure that our panelists are going to be able to answer those questions directly. So thank you so much for that also. Okay, so let's begin. So now first question for Dr. Prashant sir. The question is, what are some common machine learning techniques which are applied in genomics?
Am I audible? Yeah. So typically in any genomics, if we take the data is the most important aspect, right? Like, the advantage with the genomics is like DNA sequencing is happening rapidly, and we have the genome information of almost all the organisms. So you name it, whether even the organism is visible or not, the genome sequencing is being done. So we have a huge data available. Based on the data that is available or the what I would say labeling, annotation. If we have a proper annotated data, then we can go with the supervised learning. But there are many cases where the sequencing is done, but annotation is not done. Like hypothetical genes. Then we go for the unsupervised learning. So we just want to know like what are the genes that are getting expressed, or even if we take a comparison of the diseased genomes, then also we cannot really go for applying the supervised learning. Again, what are the clustering we got to apply and then get to know. So it's majorly with the data. If it is like gene expression studies, differential gene expression studies, if we do not have a particular marker to identify the gene, then so unsupervised learning is the only option left. If we have a detailed, like a well-established disease, then we can go with the supervised learning. That's the option. But it goes with the, again, the patient's data, how much data is available, how much it is characterized, the disease information. Based on that, we can select the respective techniques.
Thank you so much, sir. Now, second question, Dr. Bachwani ma'am. What are some challenges associated with implementing artificial intelligence and machine learning in biotechnology?
Yeah, so the challenges can come from various sectors. First, I will talk about the challenges that we face with the data. So, application of AI and ML requires huge amount of data for deep learning. Okay, so the availability of that huge amount of data may be challenging. Together with that one, see, this data in biotechnology may come from various studies. It can come from safety studies, it can come from genomic studies, it can come from clinical trials and other things. So what happens is integration of these different formats becomes a little challenging. So that is one of the challenges that the scientists have to really work with. Coming to the technical challenges, what happens is in biological system is very complex. Anything. And the AIML, when we use particularly a lot of deep learning algorithms, they work like a black box. So we are not sure what exactly is happening inside. And as the biological systems are very complex, they are not very straightforward. There is a lot of non-linearity that is associated. So unless and until we understand what is happening, the interpretability of the model, sometimes it becomes challenging to trust what the model is. So that is one of the aspects in the technical domain. Also, if we have large data sets, the complexity, the time complexity and the computational power requirement is huge. So that is also something which needs to be addressed. These are the technical sides which are there as challenges. Coming to ethical and societal challenges, there are a couple of them. When we talk about application of AI and ML in biotechnology, we are actually dealing with very sensitive data, genomics data, protein data, clinical trial data. So these data are so sensitive and there is a lot of privacy that is required to be maintained. So that is something, and what happens is a lot of different countries across the world have different data privacy protocols or rules. So to scale it to that level worldwide, it becomes a challenge because you have to cross those boundaries to get the data. And
The last thing that I will tell is, there are, at least till now, there are less people who are experts both in biotechnology as well as in the AI/ML field. So that lack of expertise also is something which there is a huge requirement where people need to be upskilled and so that they understand both the concepts. It is not only biology, as Madam was telling, Shankarajan was telling, that it requires hand in hand, so that expertise is very, very important. One person who understands or a group of people who understand both things together. So these are the few challenges that I feel, and definitely, but as definitely technology is progressing and as human resources are also getting upskilled and others, is highly controlled.
Thank you, ma'am. Now I request Dr. Shanta ma'am to take the next question. So the question for you is, how can AI/ML assist in the development of diagnostics tools?
As I've been saying, AI plays a major role in any of the biotechnological field, especially in the healthcare domain, right? The ML algorithms can help us, given a data, it can help us for data augmentation, data labeling, likewise the data clustering, depending upon what kind of data and how huge it is, or how noisy it is. We can decide whether a supervised learning algorithm can be applied or an unsupervised learning can be applied. Right? Also, given a data, in biotechnology, the major challenge would be the data set itself, right? Getting a real-time data right from the hospitals or any of the associated organizations is a challenge, right? It is not in an open source, it is not easily available as we have for other domains. Right? That is, and along with the data set, the one more factor that plays a role is the environmental factors. So only looking at the data set, we cannot judge, apply an algorithm, ML algorithm and come out with the results. What are the associated environmental factors which leads to that kind of data set also need to be very understood in detail. So basically domain knowledge is a must, right? To apply any AIML algorithm, whether supervised or unsupervised or reinforcement, whatever it is, domain knowledge is the key. Without that, I don't think we can proceed further, right? And today with all the various chat GPT and other tools in place, right? Programming is no longer a challenge. The challenge is having a good data set and understanding the data. I should understand what does this mean. If there is an image, I should understand whether this is supposed to be a favorable image or unfavorable and to what extent. So that domain knowledge is a must. Then AIML tools, algorithms, techniques definitely add some to it, right? And then we can come up with a good results, inferences, which can help us for the betterment of the society. Come up with faster diagnostics, right diagnostics at the right time, and then accordingly the treatment can be proceeded further.
Thank you so much, ma'am. Now we'll proceed to Dr. T.N. Sadhu, sir. Can you hear, sir? Yeah. So the question for you is, what is the significance of AI in predicting protein structures?
Hi. See, these AML models, if I just take AI models, so this basically helps to predict the drug discovery, and then the AI models like AlphaFold, and then predict protein structures basically with high accuracy. So with high accuracy, if you could predict the protein structure, this will help analyze the amino acid sequences, and this will definitely accelerate the biological processes. And then I was also just reading somewhere, this will also reduce the experimental cost. See, if we could reduce the experimental cost, then I think we should go for the AI models to predict such drug discovery, and we can also work towards the drug discovery. And then this will also solve the protein folding problem. That there are different protein problems which are there, and then this can also solve such problems. So AI, I think we should look for AML models to work towards the protein, predicting the protein models.
Thank you, sir. Now moving on to Ramakant sir. Your question is, what is the role of AIML in biotechnology? I guess this is like a more general question.
Yeah, thank you. So already my colleagues and other experts have talked about the role of AI/ML in biotechnology. So as far as I'm concerned, the AI/ML, artificial intelligence and machine learning, will play a very crucial role in advancing biotechnology in several ways. So as I was looking through, AI/ML can be used for drug discovery and development. So because using these AI/ML algorithms, we can analyze vast amounts of biological data, and just as Shanta Madam was telling, the problem is data set availability. So if you have the proper data set, so we can analyze the vast amounts of biological data to identify for potential drug candidates more efficiently than traditional methods, and also predicting molecular interactions, and also optimizing the lead compounds, and also even repurposing existing drugs for new applications. So this is with respect to drug discovery we can do. Similarly, AIML can also be used for genomics and personalized medicine also. So today people are going for personalized medicine. So this AI can help in analyzing genomic data to understand any variations in the genetics or disease risks, and also personalized treatment options like that. And even ML models can predict patient responses to specific treatments, maybe based on some genetic profiles, proving precision medicine, etc. So for all this, we require huge amount of data. Similarly, there is a lot of applications of biomedical imaging and diagnostics. Like we have so many diagnosis centers where we are collecting a lot of MRI data, CT scan data, etc. from the pathology. And today the problem is the privacy of this data is very important, because as we are not supposed to disclose the privacy information about the patients. So otherwise, if the data is available, AI/ML model can be used. Similarly, we can use it for protein structure predictions, bioprocess optimization, bioinformatics and data analysis. So because now AI tools are used extensively in bioinformatics for managing, analyzing, and also interpreting complex biological data sets, maybe including genomic sequences, protein interactions, and metabolic pathways also. The next another application is like drug repurposing and combination therapy. So we can use AI-driven approaches that can identify existing drugs that may be effective for new indications, or maybe for predict synergistic effects of combining different medicines. And finally, natural language processing, as I was telling in the beginning, NLP, and we are talking about large language models. So we can use NLP in life sciences also. So NLP techniques enable mining and understanding vast amounts of scientific literature which is available, and also that will accelerate the research and identify relevant information. So overall, AI and ML are revolutionizing the biotechnology by accelerating research, also improving the accuracy in diagnostics, because that is very, very important, because we are working with the life. So the diagnostics should be very accurate. And for that, drug development, and also enabling personalized treatments tailored to individual genetics. So that's what from my side. Thank you.
Thank you so much, sir. So, sir just touched upon personalized medicine, and we all know the big noise that it's making today. So, I would like Prashanta sir to just add on a few more points with respect to how AI is being used in personalized medicine.
Okay, so personalized medicine is a kind of, it's not like a new drug will be developed for a individual person. Okay, so people should not think like personalized medicine refers to one medicine for one person. No, we have lots of medicines that are already available. In personalized medicine, let's say if we take genomics for the personalized medicine, okay, one can get the genome sequencing done for a patient, individual patient, and get his genome data, analyze his genome data. Then when we compare his genome with the reference genome, then we will get to know like what are the possible mutations that has happened in that particular patient. Now once we know the type of mutation that has happened, then it becomes easy to prioritize the drug which will work for this individual, which will not work for the individual. So drug resistant problem will be solved through this personalized medicine. So we will basically save a lot of time because the patient need not go through like 4 days, 5 days, and then come back to the doctor saying that this drug is not working for me. Again, the doctor should change or give a new antibiotic. So all those problems like drug resistance can be solved, and treatment will be efficient. Then the one can fix the drug dosage also based on his genomic data, how much quantity of the drug dosage, treatment will be efficient. And one more advantage with the personalized medicine, like if we go one level higher than just the individual, let us say if we get the genome sequencing done at a, let's say like South Indian region, not just at one individual, like South Indian region, the drugs which are working for Western countries may not work for our Indian population or let's say the South Indian population. So if we do the genome sequencing analysis of South Indian population against any other countries, then we will get to know like what are the possible changes in our genome is compared to the Western. Then we can also say like why it is working for them, why it is not working for us. Then these are the advantages with the personalized medicine or overall with the genomics. So a specific group, if we take and analyze, our effectiveness in the treatment will be very high. That's the advantage with this personalized medicine.
Thank you so much, sir. Over here, I would like to mention that our club, Genesis, recently registered iGym team. So iGym is a synthetic biology competition which is held and it's an international event. So it will be really helpful, ma'am, Shantarangaswamy ma'am, if you can add upon how AIML is being used for synthetic biology so they can also use it in their project.
What exactly you mean by synthetic biology?
Synthetic biology in the sense like we are trying to express something which is not naturally present in a microbe or in any other organism. So it's more like it's being synthetically being produced, though it's not completely chemical.
The synthetic data. Basically, the data is synthetic and AIML need to be applied on that, right?
Yes, ma'am, you can go ahead with that. Yeah.
We can apply AIML onto the synthetic data. But the challenge I personally feel the challenge may be with respect to the validation of the results. Right? This is true in case of even if the data is real. Right? You discovered a drug or some results and inferences. How do we validate it? Right? We cannot test it on human beings directly. Right? So that validation is a real challenge in biotechnology field. Right? Could be so that is where the guinea pigs and all are in picture, where we test it on some animals and that before going to the human beings, before applying it on the human beings directly. Right? So this challenge becomes more when it comes to synthetic data. When the data itself is synthetic, we are not very sure whether even after testing it on animals, is it going to work on the human beings as well. That could be the real-time challenge.
Thank you so much, ma'am. Now I would like to move on to Vaishali ma'am once again. So the question is, how is AI used in analysis of clinical trial data?
Yeah. So clinical trials, there is a lot of scope for AI and ML to help in the clinical trials. It starts from recruiting the patients actually. So if we take patient data, like the demography and the genetic build of that patient, and if we can have a lot of data from the unstructured data that is available from doctors' reports and other teams using NLP techniques, then we can predict which are the people who has the probability of reacting or giving response to a particular. So that way we can pick proper people or to some extent some some screening can be done on the patients that we are putting for the clinical trials. That is one thing. Then if we can churn out the data that is already available for these patients, then their physiology and their structure, then we can predict how the drug molecules will, interact with that system, and we can predict the output of the trial to some extent, provided we have a lot of data from various categories of people. That is one thing that we can try, and the AIML can come into. Then, using AI and ML on the go, means as and when we are having the clinical trials, if we can see what is the output and then push it in the training process, then we can adapt that protocol on the go using AI. So that is also one area that we can use. And simulating, clinical data. If we have a lot of data, then simulation of synthetic data also is possible using AI. But once again, the validation is a challenge, so that we have to check. And so these are the points that I can think of. And also visualization of the clinical data using AI and ML tools. That is also a very important task. Because when we have a lot of clinical trial data, we will be jumbled up and might not be able to find out the pattern. So if we use AI and ML for visualization, then what happens is the patterns come out much easier. So that is also a very important area which can help biotechnology science.
Thank you so much, ma'am, for giving us insight of how it's being used for clinical trials. So now moving on to Dr. Ramakant sir. So are you there? Okay, we'll just move on. So now I request all three of you to just give some brief idea of what projects you are doing. It would be nice if you can take some 2 or 3 real projects, like real-time projects that you're working upon, and explain how you're using AIML and how it's linked to biotechnology.
Okay.
Yeah, so as I was telling during my introduction also, I was actually director of basic studies in our university. And my area of work is mainly, applied sciences. So that is where I am using AI and ML right now, particularly pinpointing and applying it for digital science research, where we can find out new material for various kind of applications. This is one of the areas of research where most of my time is going now. And together with that one, I do a lot of work with ISRO through some clients, and where I use AIML for predicting satellite trajectories and predicting, how in the astronomy region also, how black holes burst and what is the prediction after how many years black holes will be burst and other things. So these are the areas where I am working on, basically application of AIML in applied science domains. So that is, those are the areas where my research projects are actually currently going on, and mostly with ISRO, and also I have been asking for some biotechnology related work also where I am going to be sharing. But that is still in that point.
Thank you so much.
So Dr. Ramakant here. So we are working on a computer vision project. It's not related to biotechnology. It's related to textile industry, where there is a huge demand for counting the number of threads per inch or per particular area. So we have developed a product based on using a Raspberry Pi and Arduino Raspberry Pi, and Earlier, manually they used to count the number of threads per inch using a lens manually. It was a very tedious work. Now our students have developed a product and using AIML algorithms, so we are able to predict the number of threads. It's almost 100% accurate we are getting. And yesterday only we got the project also for consultancy, about 18 lakhs we got it. The product is ready and the company is marketing it. They want 10,000 units of that. So they are going for patenting laws. So like that, AIML is not just restricted to biotechnology. And we recently completed one project for a dentist. So the dentist wanted an software or an algorithm to be developed where he wants to find out the distance between teeth and there is bone. So whenever they do teeth transplantation, it should not injure the that particular canal. So our students, we using the MRI scan data, we are able to develop the software. We identified the distance between the canal and the teeth, so that when the doctor is performing the operation transplantation, they should not damage the canal. So successfully we completed the project and we deployed in the dentist, and he is using it as a trial basis. So like that, even though students do not have knowledge of the that particular biology kind of a thing, but we still completed the project by applying the AIML algorithms on the medical field. So this is just one example of what I'm doing. Similarly, we did a project for extramural research where we using ML algorithms, we find out the short-term wind speed prediction. This is very important during satellite launch or rocket launch, wind speed. So this was a DRDO project we completed by applying the support vector machines and various ML algorithms. So today there's a vast any field you take, we can apply AIML algorithms. So like that, we have worked on many projects. Just samples I have told you about that. Thank you.
Thank you so much, Prashant. Uh, like I'm more into biological aspects. Okay, basically I'm a biologist, biotechnology, or a bit towards bioinformatics. So AIML anyway, it's a new domain or discipline. We started a bit. I'm not like I have really applied AI ML, but we applied a bit, and I will also give you a scope where you can apply. Okay, because it's a real-time problem for me also. If somebody is interested, they can also be a part of this particular project, whatever we are working with. So one, as I mentioned earlier, we are working on gut microbiome to activate some free fatty acid receptors. I will just tell you how it all started. Because of our poor diet, right? We do not have a rich fibers in our diet. So we need two proteins which are different, like FFAR2 and FFAR3. These two are important proteins. If they get activated, all the process in our body, like immune system, pancreatic cell production, all those things happens correctly. Because of poor diet, the small chain fatty acids are not produced sufficiently in our body. So this project is in collaboration with University of South Florida. Okay, we are working together almost 4 years. So we have worked out for one compound. We are basically working towards identifying the natural compound which can activate. In other words, we want to tell like if you consume these type of foods, so these are having small chain fatty acid kind of a ligands which will activate these proteins. We are successful with the FFAR2, one of the protein. We identified fenchol, which is there in basil plant, tulsi, and we published that work also. Then we started like 1 lakh compounds, 1,44,000 ligands. But currently we have around 4,80,000 ligands. And you all know that performing molecular docking of this 4,80,000 ligands is a very tedious process. So we are working towards how best we can apply this AIML to reduce that 3,80,000 compounds to 20,000 or 40,000 compounds. So currently that is what we are working. We are interested in identifying the pattern and the key functional groups or scaffold by which we can reduce the count from this 4,80,000 to somewhere around 40,000 ligands. So if somebody is, like as I mentioned, biologist, so if somebody is really good in AIML and want to collaborate and then say like, this is how we can. The University of South Florida, they will do the wet lab validation. I got to tell them the yield molecule. From this point, this is the yield molecule you test. Because for the membrane proteins, testing here is very difficult. So they do the testing, but I have to tell them the compound. But screening, this is one of the major challenge I am facing. Again, it's a open question. If somebody is interested and then come up with a plan saying like, like small chain fatty acid like acetate, butyrate, propionate, the similar patterns, they naturally agonists. These are the training set I will use. And in your data set, these are the mapping points. I can reduce like this. This is how the accuracy will be. If you can demonstrate and work on this particular project, I'm more than happy to include anyone into this one. Okay. That's what I am working on. Thank you.
Thank you so much, sir.
I have been working on various consultancy projects and funded projects. The few of the projects which I have completed were from Karnataka Forest Development was the first project which I had undertaken. After that came the DRDO project from the CABS lab, Center for Airborne Systems. Right? So they had given us 3 modules to be developed with respect to the project management, quality management, and the workflow management. Right? So that was a very challenging project since it was real time and the lab is right now using the product that which we have done into their day-to-day activities. Right? So for every day-to-day activities, planning, meeting plannings, and how the files move from one table to the other next table, everything is automated now, and that is where the everyday life has become simpler for them. So that they had appreciated this very much at the end of the project. Right? The other ongoing project that I could like, I cannot give all the details, but the glimpse of it. The one more project, DRDO project, which we are undertaking, which we have taken up and almost at the verge of completion, is from LRDE labs. Right? So there they have a lot of satellite images, and we need to analyze those images and come up with, as we know, satellite images are very clean images taken from the satellites, and SAR images. With the images also, we are working on SAR, synthetic aperture radar images, which are very clean images, so there is no noise as such. But still, identifying the objects in those images is a challenge. So object detection, tracking, and then coming up with the analysis. That is the project that we have taken up and almost at the verge of completion. The next project which I am working on is from ISRO Respond project, which was sanctioned recently. That is also on satellite images, but here we have to play around with the optical images. The first one was the SAR images. This here it is an optical images, and since it is an optical images, we do have a lot of noises here. Right? So data pre-processing is a must, and again after pre-processing we need to do the object detection and tracking. Right? So basically they have given us a sample of ship object detection. Right? Ship objects detection. So given any images taken from the satellite from the ocean area, we will have to separate it whether it is a land or water, and if at all it is a water, then is there a ship, any object, and is that object a ship? If yes, then is the ship stand still or is it moving? And if at all it is moving, in what direction is it moving? And at what speed it is moving? All these things we need to analyze using AIML algorithms. That is the ongoing project currently. Hopefully we may get more projects in future with collaboration with Biotechnology department. Thank you.
Thank you so much. Thank you so much, all panelists. It was my pleasure moderating and hosting all of you for the session. It was really very insightful, and I'm sure all the participants also have taken key takeaways. So right at the outset, we started where the panelists told us the importance of how CS and bio go hand in hand, and it's not going to work in isolation. And of course, I'm sure none of us are going to forget the analogy of Anna Samvar, so it's going to be very easy for us to remember that. And then we also did discuss about various challenges in application of AIML in biotechnology. And one important challenge, which I'm sure all of us as students can basically focus upon, was the lack of domain knowledge. So that actually offers a unique advantage to biotechnology students. So it actually offers a unique advantage to biotechnology students. It's a message to all of us that we need to really work on getting our domain knowledge, much more stronger, and that will actually help a lot when it comes to application of AIML in biotechnology. And again, coming back to the panelists, it was really nice hosting all of you from such different disciplines. Some of them working on textile, material science, gut microbiome, to projects with DRDO and forestry. And you also covered a broad range of topics of how AIML finds application in genomics, proteomics, clinical trials. So thank you so much, respected panelists.
Thank you so much. Thank you, Dr. This really enriching session for us also, listening from Madam, Sir, and other panelists from the who have joined online. It is really a very
Thank you for the opportunity.
Thank you, ma'am, for the opportunity.
We had an opportunity to listen to the aspects are well taken, and the students and the faculty over here, and also the online participants. I want to address them also, so they are also listening. So you may, if anybody comes with your expertise, students, I will the collaboration.
I think Vidya ma'am is already helping us for one of my research scholars with the data set.
We are there always, like it's only collaboration which will isolation will never.
Thank you so much. Thank you, Ramakant sir, also for joining online.
Thank you all.
I request all of you to join Mangala Mandapa 20th and 21st. This is just a pre-conference, so please come and you can please encourage your students to join. It's definitely free. And maybe if they want certificate, I can share please share. I will share that. Since it is going to be very good, I would request all the students to encourage, especially all the people working in this panelist, please encourage the students to come. You are to invite actually, because of the time slots which is to happen there, and I think science gets diluted in the event. That's the other reason I thought. Yeah, so it should be only the scientists and the students to have focus session, because the event becomes more on other things. So I thought then I took this decision. Students who are okay with us, and RBC definitely let's need to do that. Thank you so much, but it is unfortunate we cannot have Ramakant and Sagar, though they are in the same campus.
I request Vidya ma'am to keep the sublinks to all these panelists. Mr. Shankar sir.
Thanks for coming. Please come for the conference.
Bye, Shani ma'am. Shanta ma'am.
create more opportunities and we want to work more. So that's the reason for
Thank you so much. We have arranged for tea and coffee outside, so please all of you join us. And we'll be starting with the next track, that is healthcare, by 11:40. We also would like to introduce Dr. Shubha ma'am, who is joining us online. Ma'am, are we audible? Ma'am, you're not audible. Shubha ma'am, can you please mute and talk, ma'am? We'll just check if you're audible. No, ma'am, not audible. Ma'am, is it possible that you leave the meeting and rejoin, please? Can you please turn on your video also, everybody's seeing, and also the session is being live streamed on YouTube, and we have 600 plus participants who will be viewing the entire panel discussion. Okay, so meanwhile, we can begin with the panel discussion. So we'll start with a short introduction of all of the panelists. The panelists, could you please introduce yourself?
So I hope everybody knows my name. I don't need to tell that again. Yeah. Okay, for you to all right. So I'm Dr. Sumatra, and I work as an associate professor in the Department of Biotechnology.
Hi, myself Rahul. I'm from Biotechnology department. I'm working in Biotechnology. Thank you.
Hello, this is Dr. Trilok Chandra. Most of you will not be known. We are all from the first year in CTG. I am from industry background. I work in the pharma industry as well as biotech industry. And vast experience in terms of academics as well as industry. So I'm happy to be in this panel.
Hello everyone, good morning. My name is Dr. Pradeep Kumar Deshar, and I'm working here from last 17 years. My core area is pharmaceutical technology. Last 17 years I'm working on a lot of research paper and the patent we could file and we could grant also. I'm very happy to be part of this. I'm very happy to take your questions. Thank you.
Hello everyone, this is Lord, Department of Microbiology from Sona College of Arts and Sciences. Thank you.
Okay, so let's begin with the panel discussion. So one question will be posed to each of the panelists and will be collected by the students.
So let's start with Dr. Praveen Kumar Gupta, sir. So the question to you, sir, is what are the most promising advancements in personalized medicine, and how do you see pharmaceuticals and the industry being integrated with personalized medicine, sir?
Yeah, so actually it's a very good question. So yeah, I think about the personalized medicine, if we talk, there are the different perspectives to the different people. Most of the people they think like if I take for example like one paracetamol, which is 500 milligrams for everyone. People they think like if I take 650 milligrams, so this will be a personal medicine for me. But actually, like personalized medicine, it is not like just based on the dose or delivery of the drug. It is actually based the thing because certain people are coming from a different background. They are historical. They are having a different history perspective. They may be allergic with certain kind of things. They may be having certain kind of the drug side effects. And apart from that, in fact, their own family, maybe someone is, for example, like second or third generation, if nicotine addiction or alcohol addiction, this is going to play a lot of role overall, even it is coming second or third generation of the person. So personalized medicine, I would say, like first we should have the complete data about a person, mostly relevant with the historical records, mostly relevant with the allergy kind of things. We must be aware what kind of like habit he is having. Either he is like vegetarian, non-vegetarian, either he is addictive towards certain kind of consumption of product. Okay, so this is how like we can design or we can frame a medicine which may be a single medicine, which may be part of medicine, which may be combination of 3 or 4 medication. So and about the like market prospect, I would say I think India is one of the major market for pharmaceutical industry. Everyone is looking for India because we are having cheap drug production. In fact, comparatively, our paracetamol, I think nearly maybe 1 by 50 times or maybe 1 by 70 times lesser than costing if you are living in UK. So I would always appreciate if this kind of opportunities are growing much better in India.
The next question is Mr. Dr. Tilok Chandra, sir. Sir, how is precision oncology evolving with the advent of new biomarkers?
Yeah, basically we all understand a lot of cancers is being spread in various modes in terms of different cancers are available. Now these cancers have different characteristic features, properties which have been exploited in terms of targeting these particular cancer cells. So most of them, whatever is happening is like the cancer will spread and then the patient is not. The problem here is like we are unable to locate what exactly is the cause for these type of cancers now. So with the advent of proteomics and genomics in place, so we could exploit these two subjects in terms of finding out the new biomarkers, wherein these biomarkers can be easily be targeted these particular cancer cells. Right? This is what I understand from this.
So the next question would be to Ravi. So the question is, what are the latest breakthroughs in immunotherapy for cancer treatment?
Actually, before going to tell about these, the breakthrough of these cancer therapies, I wanted to highlight some of the information regarding the cancer treatment as we are doing in the health sector. Because why I'm highlighting this one, I'm from medical background. I have faced so much of the patient with the cancer patient. So first we have to rule out whether which type of cancer with respect to the type of the tissues, organs, all those things. So in the science and technology, both the things have to be working hand to hand. So in the clinical aspect, we are going to be understanding with respect to the etiological factor, the positive factor behind it, cancer genesis factors, The next level, these four level of evaluation of a patient, it's going to be evaluating within 30 to 20 minutes of the evaluation of a patient. The first thing is etiological factors. Second one is pathogenesis. Actually, I'm asking some of the inspection, palpitation, and interrogation of the three types of the examination. We are going to understanding the pathogenesis. The second level is probable diagnostics and differential diagnostics. So both the way we come to a conclusion of the type of the disease of cancer. We ruled out of the non-communicable diseases of the cancer. The fourth one is the treatment. So in all the four levels, we are going to be having the outbreak through the technology, science and technology. Both the way they have to be developed with the technology with all the levels of evaluations. The structural functional unit of the life is the cell. So forward movement of the cell is having the tissue, organ system, organism. The backward movement is the major, the important is the science development. So biomolecular level, atomic level, cellular level. So cancer, we have to be developed in all the aspects of ionic level. From ionic level to the organism level, we should have the data, information about the particular patient of the treatment before going to have the treatment. So with respect to the question, after the question of the immunotherapy, we can't wait for that, all those things, because we want to make some kind of the outbreak of technology based. The first thing is immunotherapy outbreaks of this. After getting the therapeutic of drug molecules, Aushadha and Manasika factors. These are the four factors. If it is not possible to treat, we have to see the very advanced type of the things is CAR T-cell therapies, cell-based therapies. As told, the personalized medicine, and quick outbreak with the biomarkers, and also very targeted the pathogenesis, targeted things, and reversing the process of the pathogenesis. That is the one major targeting, identify the pathway of genesis of the pathways, and how to inhibit the formation of the pathogenesis. Okay. So next, these are the clinical application, bottom-up approach of our treatment. But because of huge population, because of diversified the mechanism of pathology, we cannot wait for the bottom-up approach. We need to see from the top to bottom approach of the treatment. So here we require the computational knowledge, technology-based electronics knowledge, image processing. As before the panelist they're telling me, the movement of the ship, how the movement on the water, direction, time, speed. So these are all the things we have to be move forward. So science people are going to be thinking in lower than before the cell formation. Ions, atoms, molecules. So these are all physics, chemistry, mathematics people are going to be working very extensively. So in continuation of that, we biotechnology people are going to be taken care with the cellular biology or cytology. Next level is immunohistochemistry. So that is the one is going to be confirming whether these cancerous tissues are going to be having a cancer or not. Next level is probable diagnostics. They are going to be mimicking the signs and symptoms. They are going to be mimicking. With a different versatile the signs and symptoms, you have to be ruled out. Come to a conclusion. We don't have the time. We don't have the patience to treat. The patient itself already is gone with the money, with the mindset. Minimum time, minimum effort, maximum health. Two things are there. One is maintenance of healthiness of the healthy person. Second one is curing the diseased person. The first level is maintenance of the healthiness of the healthy person with the personalized the educations. Okay, that's the one. I'm reversing the pathways. Next, the checkpoints. In every level of immunotherapy with the cancers, every level we need to take a checkpoints and find out what is the cause behind it. Cellular level or tissue level, organ system level. In all the level, we should have a checkpoint. I will take one example of the cellular level. What is the cell cycle pathways? What is the epidermal growth factors, vascular endothelial growth factor receptor? So many factors are there. We need to find out the way of targeting in very fast way. Then conversion into digitalization, standardization of the protocol, standardization of the protocol for next coming up people, making everything in triplet, three times, 4 times with the standardization of the protocol. That too, having the natural materials, semi-synthetic materials. If we are not getting the natural material or semi-synthetic material, we have to go with the synthetic materials. And drug delivery systems, improvisation of drug delivery systems, internal medicines, external medicine, psychological support by himself, and how the support with the family members, how the family with the societal support. All these things are very much required for the controlling, regulating, moderating cancer. Thank you. I hope.
Thank you, sir. Just to hold the mic closer to yourself. The next question is to Dr. Sumatha. What are the most significant lifestyle disorders currently affecting you, and how have these trends evolved over the recent years? And also an add-on question, if you could suggest a few ways to fix those issues.
Okay, good morning, everyone. So the question is about the lifestyle disorders that we have, especially among adolescents. I would like to address. Am I audible? Yes. So especially among adolescents, right? So now to name a few of the disorders, the first in this list is obesity. Okay, this is followed by cardiovascular diseases, and then you have diabetes mellitus, you have mental disorders, you have substance abuse disorders, you have sleep disorders. So many disorders can be listed. Now, out of these disorders, obesity is a major concern for us. Why obesity is a major concern? I'll come to that in a minute. So now, obesity, if you take the trend compared to 1990 to 2020, in 1990, the number of obese people, right, the adult obese, the number of adult obese people have increased or doubled when we compare to 2020 data. But for adolescents, it has been quadrupled. And now obesity is not just about appearance, or obesity is not just a matter of excess weight. Obesity is a metabolic disorder. Okay? So now when it is a metabolic disorder, what happens? It disturbs your metabolic pathway, and what it does is it produces a lot of inflammatory cytokines. And these inflammatory cytokines have an impact, and it can lead to inflammatory disorders. Now, the list that I have given you in the beginning, right? So I had listed out, leave the mental disorders and leave the substance abuse disorders. But the other disorders that I have listed, they all are related to obesity. Okay, so take for example, cardiovascular diseases, hypertension, diabetes mellitus, even mental disorders, because lack of self-esteem may be associated with that. So obesity is one of the important factors, and it needs to be addressed. Now, when it comes to adolescence, what happens? And why adolescents should be taken care, especially in this matter. Now, what happens in adolescence is, there is a growth pattern for all of us, right? So when you are growing up, the hormone levels keep changing. So when the growth happens, the cortisol levels keep changing, the growth hormone levels keep changing. So now what happens in adolescence is, the secretion of insulin is one thing, and then when insulin gets secreted, it has to be taken up by the cells. So that is the second thing. Now, for example, for us to understand how obesity actually plays a role, you need to know how, especially in pancreatic cells. So we all know in our body, only beta cells of pancreas are able to synthesize insulin. No other part of your body can synthesize insulin, right? So now what happens over here, when you take a food, so for example, you've taken your breakfast and you've come. The body has processed it and it is converted to glucose. Now what happens? In response to the increased levels of glucose, your body, your pancreatic cells should be able to secrete insulin, right? So now how does the beta cells understand that the insulin levels has to be, kicked up in the body? Which means it has to recognize the glucose increase in your blood. So what happens is, in our cell membrane, all the cell membranes, there are different types of receptors that are present. Now in insulin cells, the beta pancreatic insulin cells, you have receptors which take up the GLUT2 receptors are very specific, which will allow the movement of the glucose from outside of the cell to inside of the pancreatic cell. Now when the GLUT2 allows, and this is a passive movement, so when GLUT2 allows the glucose molecules inside, what happens? Glucose goes up. And what obviously has to happen, why do we even take our food? We need energy, correct? So now, where is the energy coming from? The energy is coming from mainly your macromolecules, which are broken down to smaller particles. When it is carbohydrates, it is your glucose. When it is proteins, it's your amino acids. When it is lipids, it's your free fatty acids and glycerol, right? So now what happens? The glucose level goes up and the metabolic reactions should start inside the cell. So the first metabolic reaction that will happen is increase the glycolytic enzymes. So it goes through several pathways from glycolysis to TCA cycle and then oxidative phosphorylation, and then eventually why all this has to happen? Because we need ATP. Without ATP we can't do anything in the life, right? So ATP has to be generated. So now when ATP is generated and the ATP concentration goes up inside the cell, now what happens? There are potassium channels in your beta cell receptors, right? The beta cell membrane. So now this potassium channels will be blocked in response to increase in ATP, because the body has to sense every minute what has to synthesize, right? So body is a perfect machine, but that we are given it, and it is the most expensive machine, right? So now what happens? This potassium channels will close, and in response to this, calcium will be getting inside your pancreatic cells. Beta pancreatic cells. Now once calcium moves, it triggers the release of insulin, and insulin goes out of the cell. So this is the first step, right? So after the ingestion of food, what happens to your cell and how insulin is released. Now the second part to this is, now insulin has to be recognized by other cells of your body, right? That is where the problem actually happens. And what happens over here, once insulin is secreted, we all know insulin is a water-soluble hormone. So they cannot enter the cell directly, right? So that means insulin needs receptors on the cell membrane. And there are two important receptors for insulin, which is IRS1 and IRS2. Okay? So now when insulin comes and binds to these receptors on the cell, then what happens? These receptors are cell membrane receptors, which has its connection outside the cell and inside the cell. So the moment insulin binds over here, it triggers a cascade of reactions inside the cell with the help of second messengers. Now when insulin binds over here, there is a particular IRS receptor which has to be phosphorylated. And you all know that proteins are made up of amino acids, and there are amino acids which have hydroxyl groups. And these hydroxyl groups will undergo post-translational modifications as and when required by the cell. And this is a specific place it has to be phosphorylated. With the case of IRS1, it has to be phosphorylated on a tyrosine residue. But if it gets phosphorylated on a serine residue, then your body cannot recognize insulin at all. Even though insulin is there, even though insulin is binding to the receptor, but if it is not getting phosphorylated at the right place, then insulin is not recognized by the cell, and then insulin sensitivity drops, insulin resistance shoots up. So this is the second step. Then so this has to happen properly, and then what insulin will do? Insulin's job is to put glucose to several different pathways, reduce the amount of glucose that's present in your cell. So what it can do is, it can convert glucose to ATP by going through all the carbohydrate metabolism pathways, or even if excess glucose that is present, which is more than the ATP demand of your body, then it has to be going to the lipid biosynthetic pathway. So that is one thing that I always tell. You don't need to eat directly fat to accumulate fat in the body. You just need to have extra carbohydrates and still it can synthesize lipid in the body. So that is why we have something called as essential amino acids, we have essential fatty acids, but there is nothing like essential carbohydrates. Because our body has the capability to synthesize glucose from other sources, just from proteins or lipids also, it can synthesize the proteins. Now coming back, the first step is insulin has to be secreted so that the receptor function should be good. And after the insulin secretes, it has to be recognized by the cell. So that is the second step. And now coming to adolescence. Now what happens? As I told you, from morning till night, there are different hormones that are secreted in our body. Right? When you wake up, just before you wake up, cortisol level goes up because the body has to prepare for action. And cortisol also, though it helps us to prepare for action, increased levels of cortisol over a long period of time can cause stress in the body and that can also cause inflammation. So now increase in cortisol will actually decrease the insulin production, and then that's how, even though, for example, for a diabetic patient, if they test their blood glucose level 2 hours after their dinner and then they test their blood glucose level in the morning, obviously everybody will think the whole night the glucose has been utilized. So that means your blood glucose levels in the morning should be less than the blood glucose level that you had checked after dinner, right? But usually that is not the case, and that we call it as dawn syndrome. Because the increase in cortisol is reducing your insulin activity and that thereby increases your glucose levels. So the glucose level usually when you wake up, because the body is preparing for action, right? So now in adolescence what happens? There is increase in cortisol levels and there is increase in growth hormone levels. Okay? And the growth hormone level is not going to be the same throughout the day. When you wake up in the morning, your growth hormone levels are different. When you do it in the afternoon, it's different. In the night, it's going to be different. So now this growth hormone has 3 effects. Okay? When the children turn from, say, 16 or say from 13 years it starts. The puberty starts at 13, right? So now at that time, the growth hormone levels kicks in, right? It just, multiplies by many numbers. Now this growth hormone has an inhibitory effect for insulin. So it inhibits insulin sensitivity of your body by 3 means. As I told you, for the insulin to be accepted or to be recognized by your cell, it has to be phosphorylated in a certain place. And what this growth hormone does is, it reduces the insulin sensitivity by blocking the phosphorylation. That is one thing that it can do. Or even if it's allowing the phosphorylation, it's going to allow the phosphorylation in the hydroxyl group of serine residues. But when it is hydroxylated in the serine residues, all the pathways that are there down the line will get blocked. Only when it is phosphorylated at the tyrosine residues, it is going to be allowing the cascade of reactions which will allow the glucose to be utilized by the cell. So this is also going to be blocked. And growth hormone also is going to increase gluconeogenesis. So now gluconeogenesis, as I told you before, the body has the ability to make glucose on its own, right? Which means from amino acid, carbon, skeletons, the body can synthesize its own glucose and liver has the ability. But usually what happens is, when you do not have enough glucose in the cell, in the body, then the body tries to get glucose from other sources. Now it increases gluconeogenesis, thereby it increases the glucose level. So that is the second way in which the growth hormone plays a very important role in increasing the glucose levels. The third way is the growth hormone increases lipolysis. Which is, what is lipolysis? Lipolysis is degradation of lipids, and when that happens, it is going to produce free fatty acids in the blood, and the free fatty acids again is going to go and block the insulin receptors, and the insulin will not be able to bind. So this is why in adolescence it's very important. See, for type 2 diabetes mellitus, there are several factors. Genetic factor contributes to a larger extent. Okay, but apart from the genetic factor, you also have lifestyle, the sedentary lifestyle that today's youth that we have. The sedentary lifestyle plays an important role, and the diet that we are taking, which is usually calorie rich but low in nutrients. Now what happens is, this contributes along with the genetic factor and along with the natural developmental process that the adolescent will have, which is the increase in the growth hormone levels and increase in the cortisol levels and many other hormones that are there. Right? So all this together, what it will do is, it will actually cause a very important significant disease, which is type 2 diabetes mellitus and obesity. That's why I said it is not a standalone thing. It is just not the looks, it's just not a matter of excess weight, but it has larger complications, which type 2 is one of the important complications, but there are many other complications associated. So when type 2 diabetes mellitus is there, then that can lead to other diseases like retinopathy, neuropathy, and cardiovascular diseases. All these will, add-ons to those things. So that is why obesity is not one thing. Obesity, even cancer is an inflammatory disease, and obesity can lead to cancer. Right? So we can't just take this as a very small thing, just the appearance is not the one thing that we are talking about. It is a metabolic disorder, and the consequences of this metabolic disorder down the lane can cause other disorders. So I think from the list, most of the diseases are covered based on just one disease itself. Right? So I hope you all are clear that obesity is going to lead to inflammatory disorders, and many of the diseases are listed as inflammatory disorders and non-communicable diseases.
So we'll move on to the next question to Dr. Shubha. So the question to Dr. Shubha is, in the recent years we've seen an increase in online consulting and telemetry and telemedicine as well. So what role does telemedicine play in improving patient outcomes now? Just a second, ma'am, you're not audible.
Shall you audible?
Yes, ma'am, you are.
According to me, we are not able to see each and every person in the village people like that. We are not able to reach each and everyone. So in that condition, the telemedicine plays a major role. So it provides a great access to the healthcare, especially in the remote areas. So by this telemedicine, we are able to continuously monitor the patient. We are able to access the progress of the patient. So I think it's the best way to reach each and every people in the remote area also. So it gives the best outcome. I think it gives best outcome and satisfactory results. Thank you.
Okay, so we'll have another round of very quick questions. Since we're a little short on time, I'd like to request the panelists to keep it as short as possible. So, sir, Praveen Kumar sir, following up on the previous question about personalized medicine, diagnostic accuracy plays a big role in transforming healthcare. So, I'd like to ask you as to how artificial intelligence also aids in the transformation of diagnostic accuracy in healthcare.
Okay, so actually I would like to continue what Shubha madam just said. So telemedicine, there is a concept of e-health, there is a concept of m-health, e-health, electronic health, m-health, mobile health, right? So in general, like these are the parameters, these are the tools, or these are the techniques. It is actually creating easy life for us. So in future, I would say, for example, like someone could not go to the doctor because there are certain reasons, and that's what the disease was not diagnosed properly. So the disease diagnosis, like as much as possible, like if we go for more technology based, there are more softwares are coming. I think we are not very far. Like with a mobile phone, we will add some, accessories or certain kind of gadgets. And then we need not to visit to the X-ray machine. We need not to visit to the MRI. We can directly put that gadget to our mobile, and that someone is sitting on 2,000 kilometers, he will be visualizing. So overall, in general, the concept is that better technology will provide the better diagnosis, and better diagnosis will go for the better treatment. That's what I want to say. Thank you.
The next question is to Jog sir. What are the latest trends in digital therapeutics for chronic disease management?
So adding up to overall things what we have learned so far in terms of diseases and how to manage these diseases. So basically, all have these wearable technologies in terms of smart watches, or wherein you can monitor all your health, right? So everybody are having like in terms of what is the pulse rate, what is the heartbeat, or whatever sugar levels in some cases, most of them are having it, right? So these are the technology which is emerged right now, and everybody are utilizing these things. So one step ahead, like recently, I happened to attend one of the workshops where they have come up with a German technology, which is known as ED, the product name ED, HEALY. So this technology, what it does is like in terms of they have a concept like all the cells, what are the biological cells, whatever we have here, if they are healthy enough, there is no disease at all. Like it does its function in terms of what a natural way of functioning the cells. The moment the cells deviate its natural way of functioning, there lies the disease what is being triggered in human beings, right? So they are trying to target those type of those cells which can be treated in terms of healthy cells, right? So for that they're using certain physics, as I told, physics, biology, and chemistry. So these are the things what are being used there in terms of the healthy cells. We are trying to trigger the growth or the functioning of cells in the body. So they are using some electronic devices which are being attached to your body, and these bodies will have devices will regulate your cells in such a way that they function normally without any abnormalities. Even there is no abnormalities, there is no diseases symptoms. So they are being tested. So this is one of the things what is happening in right now as latest wearable technologies which can be used. Thank you. Thank you.
Raju sir, could you please answer this question? So, how do we develop scalable technologies or scalable solutions to address mental health issues that are seen to be more prevalent, especially after the pandemic?
So, after the pandemic, before the pandemic, and during the pandemic, these are the three phases are there. So, before, during, and post, pre-operative, operative, and post-operative. Here the question is post-operative. So the post-operative of the treatment is depends on the operative damages and pre-operative the conditions. Pre-operative conditions, situations, what are the causative factors behind it, what is the pathogenesis, and how the probable diagnostics and differential diagnostics are there in the how the person is going to be adapted, is going to be taken the pathological the agents into in his body, the system. Pre-operative. Operative means along with him, the person. Along with him, then next up the person is the physician and then medical people, those who are getting involved in the during the treatment. That's the next threatening part. The post-operative is having the remnant parts. Whoever is having the very winning part, winning persons, to get rid of all the condition of the critical condition of the COVID situation, the faced and win. So that people are going to be only facing the issues, the post-operative. The remnant parts are there. Remnant, the materials are there during the post materials, the COVID materials, and also the treatment part. That's called as the genomics, protein, the interactomic materials are there. During the metabolic process, they are going to be having inside it. They are not yet removed. So there is having they themselves are having two things are there, no? Exotoxic and endotoxic. So earlier it's having the exotoxic, and then it's going to be having the endotoxic materials. Going to be having. So for that we need to understand very quickly, understanding the situation by taking the case history. behind it. Personalized history and family history. Okay, and then there is only geographical area history about it. And make it into a very precise form, digitalization form, documentation form. Okay, so next part is boosting to that, where there is having easy way of understanding the age, sex, regional area. Okay, so the age, especially the prone people for more the issue people is pediatric people and geriatric people. So the middle-aged people they are going to be having a winning because of their immune system, because of them they are having a adaptability is very high. But the threatening people is pediatric people or geriatric people. And then even in the geriatric people, they are going to be having associated with the other complications, the medical the issues. Already they are going to be having daily they are going to be having a BP, diabetes mellitus, the regulation of the medicines. Definitely they are going to be having the issue with respect to the bodily issues and then mental issues. And then during the COVID condition, the treatment, the first priority is prevention, making the distance, maintenance of the distance. The people definitely they are going to be getting the isolations. We all know that we are all going to be having a social life. When the person is going to be having a isolated for a long time, mentally they are going to be having a stress. So to overcome this, making a regeneration of the rapport with the friendship is very difficult. It depends on the time. Earlier time, today and tomorrow is going to be changing. The time is going to be changing, place is going to be changing, and the people are thinking, the mentally is going to be thinking is totally different. So based on the case to case, taking the interrogation questions and boosting them, we are going to be supporting in all aspects. The societal problems, societal level, family level, personal level, we should be very courageous. Adaptive engineering, adaptive technology should be implemented in every minute, every level. Then only it's possible to make it very easy. And then the digitalization of the documentation, information. As Trilok sir told, taking the data, whatever they are having the pulse rate, the personalized information is there, respiratory distress, all the data information to the stakeholders, very family doctors, family persons, very fast, and come up with the isolation in the problems. These are what you call the clinical aspect, biochemical aspect, pathological aspect. There are so many things are there with respect to the come up with the post-COVID issues. Okay, and making with respect to the boosting of the vaccines production, vaccines. Okay, so these all things are there. Hope we will going to be come up with them. We are not yet come out till now. Okay, in the rural area, in the urban area, again it's going to be the technology is not reached to the rural area people. After getting the rural area also not reaching to the last end user, the person who is going to be suffering is not going to be using in a very good manner. Okay, in a correct time. It's going to be lacking now. It will going to be taking another 5-10 years. Okay, this is my opinion. Thank you.
The next question is to Ms. Shubham. Ma'am, how is big data analytics being utilized in predictive healthcare?
Alright, so as I was talking, for example, any disease that you take, right, it is not a standalone disease, correct? So now, when you talk about a particular disease, there are certain diagnostic factors that are available, and these diagnostic factors are considered as a standalone factor. For example, if you take diabetes mellitus, there are certain markers that are available to test whether a patient is diabetic or not. Or if you take for cancer, there are certain biomarkers that are available. But only these are not the factors that are going to be involved. So now when you can have AI technology, which can actually, based on predictive analytics and based on algorithms that can visualize things and make a sense out of all the data that is available, then that becomes a very important thing. For example, when we go
for a basic health checkup, there are certain tests that are given to us. One is the basic CBC that we are given. Then there is a lipid profile testing. Then there is, all the diabetic markers, then thyroid profiling. All these are done. But for example, if a person has got diabetes, the focus is mainly only on that, right? But then when they compare all these things and then correlate and see based on the pattern recognition model, then it will be, much more informative in terms of, predicting the disease early on. For example, there is a recent study in Stanford that was conducted in 2021. What they've done is they have taken the electronic health record. There are privacy issues and other things, but it is just a data anonymously that was taken, and this electronic health records they are subjected to AI, and they were able to identify individuals who are in the zone of prediabetes, diabetes, and cancer based on very general data. Because it is going to consider lifestyle factors, it's going to consider the genetic predisposition, it's going to consider the family history, everything is going to be considered and they are predicting it. Other example is certain lifestyle disorders that we were talking about. For example, if a person is diabetic today, this disease hasn't started today, right? They probably were in a pre-diabetic phase many years back and which was not at all detected. Right now, there are AI technologies which can detect a disease 3 years earlier than it can be detected by, the traditional methods. So that is something that, the pattern recognition models and algorithms that are used through this machine learning process will be able to detect things based on not just one factor, but several factors will be considered, and pattern recognition is very, very important to identify any disease early on.
Would you please like to add on something to the same question?
Yes. As Ma'am said,
Yes, ma'am. You're audible.
so this big data analyst, it is nowadays it's a boom to the hospitals. So it is used to diagnose the various diseases, and we are able to maintain the records of the reports in the big data analyst. So it is one of the application of this. Then it is also used to predict the disease outbreak in a particular area. So if suddenly some disease outbreak in a particular area, so it is also we are able to analyze with the help of the big data analyst. Then it is used to identify if some population, if particular people, they are in particular risk, that is some infection is heavily spread in that area. So it is also monitored with the help of this. So by using this method, so blockchain method, this are also used in the big data analysis. So it is used to maintain the historical data also. So if we are maintaining the historical data, so if in future, if the same problem comes again, we are able to rectify with the help of that historical data. So it is also one of the applications. Then in the hospitals, we are able to maintain the patient's record. So in the hospital, many patients' records are available. So the manual method, it is not possible. So if we are using this big data analyst method, so we are able to maintain patient's record clearly whenever we want to retrieve that. So we are able to easily retrieve that. So it is one of the applications. Then it is also used to optimize our treatment plan. So if we are maintaining the records, so if the patient comes with the same problem again, so we are able to optimize that. So it is also one of the main advantage, that it is also used in personalized medicine also. So if we are maintaining the records of each person, so we know the patient's details. So we are able to give the medicine based on the person. So it is also mainly used in personalized medicine also. Then it is also used to improve the hospital resource management. So it has a wide range of applications in healthcare. So it's a boon to the healthcare department. Thank you so much.
That was very insightful. I'd like to thank all the panelists for being here. Over to Anirudh for the felicitation, followed by the next session. Dr. Sumatra. Dr. Raju. Dr. Trilok. Dr. Praveen Kumar.
Thank you so much, panelists. It was really an insightful session. We'll be starting with track 3 panel discussion for genetics in another 2 minutes. Esteemed panelists, I request panelists to please come on to dais. We have with us Dr. Ashwani sir and also Dr. Nagashri ma'am. And joining online with us, we have Dr. Manjula ma'am, Dr. Ravi Shankar sir and Dr. Rajeshwari ma'am. Can all the online panelists please confirm if we are audible, and also can you please check if we are audible from your side? And it'd be really nice if you can all turn on your videos. Yeah, thank you. So without any further ado, let's dive straight into the panel discussion. So the speciality of this panel discussion is usually the moderator just frames the questions, but over here we have asked our student representatives to frame the questions, so it's going to be straight students' questions being represented through me for all of you. And also I would like to inform all the panelists that this entire session is going to be live streamed on YouTube, and we have 600 plus participants from different states who will be viewing you. Okay, so first question, we have Ravi Shankar sir as well as Manjula ma'am who are short in time, so I'll go straight to them first. So Dr. Ravi Shankar sir, your first question is, how is genome editing using CRISPR-Cas and how has it changed crop improvement strategies in agriculture?
Can you hear me?
Yes, sir.
Okay. So genome editing is a new tool now. It is considered as a in vitro breeding, that you can breed the new varieties and new improved hybrids inside the lab. So, for example, there is a recent paper in Nature Genetics. It's called in vitro domestication of wild relatives. We know that wild relatives have very good traits like disease resistance and tolerance to biotic and abiotic stresses. But they suffer from poor quality of fruits and flowers like seeds. So one can improve those because we know the genes responsible for good fruit quality, increasing number of flowers, increasing branches. By editing, so they have in Solanum Panelist, a wide variety of tomato, they improved the number of branches and number of fruits, and even they improved the quality of the fruit. That the beta carotene has been improved by 500 times. And also we some in some for example in grapes, so downy mildew and powdery mildew are very difficult to find resistance for this downy mildew and powdery mildew. But by genome editing, we can induce that resistance. If there is any no vulnerabilities or no germplasm resistance, we can do that by genome editing. And also it can improve the quality and disease resistance generally by genome editing approach. And it's a very sure-shot method, and it consumes less time, and also it is very targeted approach.
Thank you so much, sir. Next question is for Dr. Manjula ma'am. So we just heard about how it's finding application with CRISPR in agriculture. So we make a shift now to human health. So ma'am, my question is, how is genetic research enhancing the effectiveness of transdermal drug delivery systems?
Good afternoon, ma'am. Can you repeat the question?
Yeah, ma'am. The question was, how is genetic research playing a role in improving the effectiveness of transdermal drug delivery systems?
Now figuring out the effectiveness in drug delivery.
Ma'am, your voice is actually breaking.
Yeah. Can you hear me?
Yeah, ma'am.
So workers can be
No, ma'am.
treated.
Looks like there's some network issue from your side. Ma'am, can you please rejoin? Okay, so by then I would like to move on to Dr. Rajeshwari ma'am. Ma'am, could you just add on to this of how genetics is playing a role in human healthcare?
Hello everybody, am I audible?
Yes, ma'am.
Yeah, can you please repeat the question?
How is genetics playing a role in human healthcare, especially now with the focus on personalized medicine?
Yeah, we all know more about genetics now, awareness has come. And the main important role is an individual's susceptibility for any kind of disease for that matter. So each one, the susceptibility is different. So we cannot make any generalized statement over here. So apart from individual susceptibility, the other contributing or confounding factors are also there, like environmental factors and individuals' lifestyle. All these are that which will trigger an individual's risk. So genetics really play a very important role in susceptibility for any disease. That's what I can say in a generalized manner.
Thank you, ma'am. Okay, moving back to Dr. Ravi Shankar here. Could you just add more info on how microbes play an important role in crop production and agriculture? And if you can just add on to some projects that you're working on in the same field.
See, the microbiome is a very important area of research nowadays. I think you may be knowing that human biome, the NIH is funding huge project, a very big project. Similarly, we have microbiomes in plants also. So, for example, to give you an example, Pokali is a salt-tolerant rice variety which is grown in highly salt-affected area in Kerala and Tamil Nadu. So, scientists have isolated some of the fusarium species from this Pokali salt-tolerant rice, and they have introduced that into the highly salt-sensitive genotypes called IR24 or Jaya. So these experiments are done in JKUK Bangalore, and so they could able to find that the increase in salt tolerance of the general varieties, which are hybrids, which are grown in the mainland area, and you know because of use of continuous use of the fertilizers, most of our soils are getting affected because of salinity. And and also they could see they are endophytes and they are transferred from generation to generation through seeds. So the students have worked on this, and these endophytes can be transferred from generation to generation through seed, and also they also impart a good amount of salt tolerance, and they help in improving the yield. That's one aspect. And second aspect is that they are also helping disease tolerance because this is cross protection. So they don't allow pathogens to grow or invade the plant. So this is one. So we have some in the Indian Institute of Horticulture Research, we have a microbial consortium called Arkham Microbial Consortium. So which is Azotobacter, Pseudomonas, and bacillus. So these three species we have made a commercial formulation. So this we have using it in many of the horticulture crops, which improves the biotic stress tolerance, that is improves the disease resistance, and also it improves the seedling growth. So how they improve the seedling growth is because of improving the nutrient availability to the root. So these are two aspects, and also recent people have shown that in many the hybrids grow better because they have shown that in hybrid they have a totally different biome, that is plant biome. They have high diversity is there in the hybrids, whatever grown in tomato or in maize, and also they have distinct microbes residing in this hybrid. And when you try to remove these hybrids, then the hybrid vigor will come down. So these are the many new areas of coming out the application of microbes in plant crop production, especially the crop production.
Thank you so much, sir. Dr. Manjula ma'am, can you please speak and we'll just check if you're audible?
Okay, yeah. Am I audible now?
Yes, ma'am.
Yeah.
Ma'am, so your question was how is genetics playing a role in drug delivery, and if you would, you can just focus specifically on transdermal drug delivery system.
Okay. So basically, I feel that the genetic markers that are present can help in enhancing the effectivity of the drug delivery system. And also, we all know that now we are in the third generation where we are using microneedles. So probably these microneedles can help in drug delivery with RNA, DNA, and other protein molecules too. So microneedles can also be used wherein we can use in the form of hydrogel systems, or it can be incorporated by coating or even by dissolving them. So there is another area where biomimetics can be also taken in account for studying the pharmacokinetic studies, and we can help in improvising and efficiently carry out with this drug delivery system through the skin. And ethosomes are also one part where we can work on with. So this is a large and diverse group of proteins that can lipids which can help in carrying out these protein molecules and peptides by drug delivery system. So I think the basis will be with the genetic markers, how they are going to interact with this, with it, so that we can work effectively there.
Thank you so much, ma'am.
Thank you.
Dr. Rajeshwari, ma'am, are you there?
Yeah. I'm there.
Yes, ma'am. Ma'am, can you please elaborate on how genetic analysis of microbes is important with respect to understanding the entire development of antibiotic resistance, especially with it becoming such a huge problem today?
You're asking about antibiotic resistance gene and multi-drug resistance, right?
Yes, ma'am.
Yeah, we are all aware of this multi-drug resistance bacteria, the emergence, and all this we are very much aware of. And we also know that these genes are that which are located on the plasmids, or even during the process of this cell-to-cell like conjugation or transformation. During these process, it's been transferred. So that is how the exchange allows. And during this process, resistance happens, and that is how it is spread across in the population of bacteria, and we are seeing so much of this resistance. And of course, apart from this, there are other factors also which are there, like this latent ARGs, like drug resistance genes which are present in the environment, how they are being spread across also. Wastewater treatments, then through atmosphere, and we humans, others are trying to spread this. So this is how there's a lot of work which is going on, and so many antibiotics are that which we are becoming resistant to them. So maybe some of these things are that which needs to be taken into consideration for this multi-drug resistance.
Okay, thank you, ma'am. Now, the next question is to Dr. Ashwani, sir, who is with us offline. Sir, so as we all know, FSSAI is becoming stricter day by day in India. So, do you see any role that GMOs or genetic modifications can play in terms of enhancing food safety and security, or do you see it detrimental?
Interesting question. Food. So when you talk about FSSAI and food security, and prime importance is health. And yes, wherever you talk about, the prime component is health in totality. And when we talk about health and being very close to the food, the first and the foremost thing that we should always understand it, yes, whether it is with respect to microbes, whether it is with respect to genetic manipulations, or with respect to genetic modifications, foods have been always a target. For example, when we talk about fortification, and when I say this fortification, there are two ways of fortifications. One is biofortification and one is chemical. Technically, chemical fortification comes under the regime of SSA. But when we talk about biofortification, which is food to food, that does not come under the regime of SSA. Interestingly, when we talk about all omics, when we talk about GWAS, and when I say with respect to GWAS, the most important thing is genome-wide association. So when we talk about transcriptomics, proteomics, and all, when we take a blending of all those components, the food becomes very critical. And when we talk about nutrition, and yes, there is a red and white flag is going on whether GM is good or bad. In India, if it would have been bad, then I'm sure GM cotton and GM brinjal, BT brinjal and BT cotton would have not been existing. And let us not talk about any other thing globally, particularly in USA, the labeling of GM foods is almost 31% of the total food. And the most prime thing what FSSI or food market has to target with respect to here is labeling. If we take care of labeling, I think FSSI or any regulatory bodies, the job will be very easy.
Thank you, sir. Moving on to Dr. Manjula, ma'am. Ma'am, can you please add on to what projects do you work on, and if you can just elaborate a little on one or two projects that you're working on right now?
Well, I'm working currently on antimicrobial resistance strains from the upper respiratory tract infections now, and also bioremediation is one part where I'm into, and extremophiles with basically the thermophilic bacteria is what I'm into. So whatever my work is based on, it is with respect to the beneficial areas. Apart from that, the multi-drug resistance characterization, so that we can work on the antibiotics and the alternative therapies. What I'm intending to work on. This is where my work is working.
Thank you for sharing this, ma'am. Dr. Ravishankar, sir.
Okay, my lab works on genomics and marker-assisted selection. So in marker-assisted selection, what we do is we already know some of the genes which are conferring viral resistance and fungal resistance like Phytophthora and nematode resistance in tomato. What we try to we'll work with breeders. We will try to pyramid these genes into single variety or hybrids using marker resistance. Here, what we do, we don't screen for this resistance. Since the marker is there, we can identify those individuals or hybrid material or parental material at the seeding stage itself. So once we identify that, we'll try to pyramid by using different processes and different models. We try to pyramid all these genes in one variety or one hybrid, and we can use that variety or the parental lines in hybrid development. That's the one aspect which you are working. And the second aspect which you work on genomics. Here we work on whole genome sequencing of some of the crops like okra, custard apple, and also on drumstick. So these are the whole genome sequencing helps to identify the number of genes and their segregation in different breeding generations and the marker trait association, all those things. Second aspect is genomics. We also try to understand what are the genes and regulators are involved in some of the diseases. For example, early blind is caused by alternate solenoid tomato. We know we have some resistant genotype and also we have some susceptible genotype. We develop mapping population in either F2 or across region. And we identify QDL regions, that is the genomic regions which are responsible for the disease resistance through marker trait association. Here we use different markers or we use whole sequencing of these mapping population. Once we know the marker trait and genomic QTLs, that is the genomic regions responsible for these particular resistance to these pathogens. So we will try to know what are the genes residing in these genomic regions. Thereby we can understand what are the genes responsible under the regulators like transcription factors or mRNA, non-coding RNA, or circular RNA. These are the regulators which are responsible for the disease resistance. Similarly, we do for some of the quality parameters in Goa and also in mango. So we improve the carotenoids or improve the vitamin C content or to improve the anthocyanins content. So there are many aspects. The approach is similar, but the material which we use is different. We may be using mango for carotenoids or vitamin C for the Goa mapping population. So there are different approaches. Here we use extensively next generation sequencing. Then RNA analysis, and also we do a lot of bioinformatics.
Thank you so much, sir. Next question is for Dr. Nagashri ma'am, who is with us offline. Ma'am, could you just explain in brief what exactly functional genomics is, and if you can just tell us a little on some projects that you have done recently.
Hello, happy day to one and all. Functional genomics is an offshoot of the entire omics technology. And when you talk about the omics technology, you start with genomics, you go till the metabolomics, where you have the intervening transcriptomics and proteomics, and they all have to integrate, and there should be a clear crosstalk among all these omics, ultimately to bring about a function wherein you study at the systems level in the name of systems biology. So when you talk about functional genomics, it involves all the four. Okay, you start the functional genomics with studying a function of a gene. So there are many aspects to study a function of a gene. You go for what is called as a forward genetics or reverse genetics mechanism. And usually we go for reverse genetics, which is pretty easy compared to forward genetics, because forward genetics involves too much of manpower. And in Indian scenario, I think we should actually rely on the reverse genetics approach, where you target a particular gene which you know and try to see, like if you try to perturb a gene, either misexpress, I better use the term misexpress, because you're trying to put the gene in wherein it expresses more, or try to eliminate the gene partially or completely, and try to see, like, if I eliminate the gene, what is the consequence? Okay, what is its consequence? It might be on a particular character where you call it as a trait, and then go back and tell that because I have knocked down or eliminated a particular gene, the effect is you are not getting a particular character. Okay, and also in functional genomics, you try to study the interaction, which is very, very important, because to bring about any function, the interaction between the DNA and the protein and protein interaction, whatever I'm telling is just the crux of it or the gist of it, it's very, very complicated, and it involves both in silico and the wet lab experiments to ultimately conclude a function of a gene. So functional genomics encompasses the entire omics technologies. So at RVC, we started with a little bit of functional genomics, wherein we try to express a gene from Arabidopsis in a medicinal plant to see whether it is taken up in a medicinal plant and whether a particular metabolite is increased. And we do see that though we are trying to express from the Arabidopsis, it is enhancing the metabolite. And also we are trying to work in capsicum, wherein we try to work on the pungency factor. So our area is to use reverse genetics approach, where we are trying to knock down 3 transcription factors through RNA interference technologies, and try to address, if I knock down the genes that are involved in pungency, will it reduce the pungency in chili? And we also worked on basela, which is a leafy vegetable, to check whether through knockdown or try to express it more, whether one of the metabolite, which is called as betalain, is increased or the level of betalain is decreased. And currently we are also working on the coffee, wherein we are doing through gene editing technology, trying to reduce the caffeine content. You'll be laughing, like, why to reduce caffeine content? Because caffeine has both it has both pros and cons. too much of intake of caffeine is bad, so people can have the feel of coffee with a less caffeine content. So through gene editing technology, targeting the caffeine biosynthesis, we are trying to target two genes where you are going for editing two genes and try to see or reduce the caffeine content. In addition to it, we are also working on microbiome analysis, especially to go for bioremediation. This is a technique which we have taken up recently, especially for water treatment. We are trying to go for the endomicrobiome techniques.
Thank you so much, ma'am. Dr. Rajeshwari, ma'am, could you also please add on and tell what projects you are working on? If you could just elaborate one or two of them.
Yeah. Actually, mainly my concentration personally is on genetics and epigenetic alterations, which are the main contributors, and to identify and characterize the genes which are mainly involved in the development and progression of human cancer. So I wanted to identify biomarkers. Now, presently concentrating for more than a decade on gliomas, and that too specifically on a notch pathway, because this particular pathway is that which is plays a very crucial role in regulating this cell-fate decisions and all. And moreover, the survival rate, if you see, it is very less, where it is only up to 1 to 2 years. So at the moment, I'm focusing on identification of the biomarkers on this particular pathway, wherein I was able to identify. And then the regulatory part is that which I had concentrated on. And also another aspect is to identify novel therapeutic targets, especially looking into the herbal extracts, because they are more safe and affordable as compared to the chemotherapeutic. So in that particular area, like presently we are working on this Bakoba, that is Brahmi, direct extract, as well as even the commercially available drugs also we are taking. And another important aspect, what we are looking for, just note Dr. Nagashri, she was telling about gene editing. So that is another area which I'm concentrating, where we are trying to regulate a few of the genes, and then finally look into the end aspect, what would be if you switch off and switch on. Mainly now we are concentrating on this matrix metalloproteases in North signaling pathway, where we wanted to check for the efficacy of fucoside A. Of course, earlier studies were on patient samples, but now I have shifted a little bit on working on cell lines, because I want to concentrate a little bit on gene editing. So these are the few areas which I'm working. The epigenetic aspect of the methylation studies were that which were carried out, and we were able to identify, of course, the few genes which were regulated, mainly because of the reversibility. So these the small drug molecules, whatever are there, if you are able to identify the target, these small molecule inhibitors are that which can be used. And if it is epigenetics, then the reversibility is that which it can be done. The alterations in the tumors, and you can restore the normal epigenetic state, so that we would be successful in cancer therapeutics. And of course, there are a lot of small molecules which are under clinical trials also. So these are the few areas personally which I am working on.
Thank you so much, ma'am.
Yeah.
Moving to Dr. Ashwani sir, would you please discuss a couple of recent technologies or techniques that are being used in genetics, like recent advancements in genetics and genetics?
It is the plethora of an ocean. If you talk about what genetics has done Talking from, it's like something, write an essay, like write an abstract of an essay. So I will try to write an abstract of an essay. See, first and foremost, as I said, food, yes. Talking about BT brinjal, BT cotton, moving to what we call it as golden rice. Coming to polymers, when we talk with respect to polymers, now we have genetically engineered polymers, which talk about the strengthening which have come from spiders. Then we have something, the plant fibers, which are very lightweight. We have come to the something called as composite materials. These composite materials, they are with respect to natural fiber composites. From that composite fiber, when we move into medicines, and if we talk about resistance, I think Ravi Shankar sir has done a huge, tremendous work, and he has made my job very easy. So if you talk from food, fodder, fertilizer, plastics, when you talk about daily requirements, if you talk about medicine, in all the context in a broader purview, yes, genetics is the one that has literally used that word, encroached it to make it to a better world. Think it over, the entire concept of green revolution is just a genetic makeup. Now there is a huge debate. Should we have another green revolution or we should have another GM revolution? Because when we talk about green revolution, we don't have that much space. Now the question is, do we need genetic revolution or we need green revolution? So now the entire responsibility is on these students who are the torch bearer for tomorrow, who are looking forward for how it can go. Furthermore, when we talk about with respect to all the wet labs, big data analysis, data are very huge, clouds are getting covered, trafficking is there. Now the thing is how we can analyze it, how you can conclude it. So if you talk about genetics, yes, no patch of work, no patch of component is left with respect to humans and around humans, including environment, which is untouched. And yes, there is much more to work. Still 0.1 to 1% has been touched. It's a very huge ocean.
Thank you so much, sir. We also had on our panel Dr. Savitri ma'am, but because of unforeseen reasons, she wasn't able to join us today. However, ma'am was really kind enough to share her answers pen down. So I'll be reading out a couple of questions that were posed to ma'am and what were her responses. So, one question that was asked to Savitri but ma'am was, what genetic factors influence protein misfolding in neurodegenerative diseases based on your current research? And ma'am's response is, misfolded proteins results when a protein follows the wrong folding pathway or energy minimizing funnel. It can occur through other mechanisms such as genetic mutations, errors in transcription or translation, problems during nascent peptide folding, and stresses that damage structures of normally folded proteins. Most of the time, only the native conformation is produced in the cell. But as millions and millions of copies of each protein are made during our lifetimes, sometimes a random event occurs and one of these molecules follows the wrong path, changing into a toxic configuration. This kind of conformational change is most likely to occur in proteins that have repetitive amino acid motifs, such as polyglutamine, such as in the case of Huntington's disease. One of the prime examples of proteins that catalyze their own conformational change into toxic form is prion proteins, which has been ma'am's postdoc research domain at NIH USA, which led to a publication in Cell Journal. Under normal circumstances, the cells have mechanisms to prevent proteins from folding incorrectly, as well as to get rid of misfolded proteins. Proteins that have problems achieving their native configuration are helped by chaperones to fold properly using energy from ATP. Chaperones can avoid the conformational change to beta sheet structure and the aggregation of these altered proteins. Thus, they seem fundamental to the prevention of protein misfolding. Despite their actions, some proteins still misfold. These misfolded proteins can be detected by quality control mechanisms in the cell that tags them to be sent to the cytoplasma, where they will be degraded through the ERAD pathway. Thank you so much, ma'am, for your response. Now I would like to pose the next question to Dr. Ashwani sir. If you can just briefly again, same question to you also, if you can just tell us one or two projects that you are currently working on.
As I'm working right now on yogurt, when I say most of them working on food and food related aspect, yogurt, increasing the taste, texture, aroma, and fragrance, and enhancing the shelf life. The biggest target is that. That's the reason every year, almost when I say every day basis, if you see the statistics projected from the Canada government itself, there is a loss of 23.1 lakh liters daily. So if you think it over how many stomachs we can feed, that is the one thing. Then I'm working on Bacilla, that is color enhancers and regulators, working on mosquito repellents. And when I say mosquito repellents, people call it as, in Indian version, as Jugaad technology, but do understand all the concepts which have come to a greater aspect have come through Jugaad technology only. And these Jugaad technologies, when you put it in a very sophisticated manner, they come with any of these jargons. Technically, whatever you talk about, yes, another component I'm working on is a matrix. When I say composite materials, what I have used in all the waste things. You tell the waste, yes, we have made it. And further, I am working on a type of fiber which is very much what what you call it as water resistant. It does not absorb water or adsorb water. The rest it does everything. So working on these few projects.
Thank you so much, sir. Next question to Dr. Nageshri ma'am. What progress have you made in improving crop resilience through genetic transformation, and how does this support sustainability?
Okay. There's a huge number. It's a huge number. It's not a single number. Like you have many transgenic and gene-edited plants which can actually tolerate biotic and abiotic stress. Okay, if I tell biotic, it's to resist the biotic stress. It might be against microbes, or it might be against fungus or virus. There's a huge number, like it has been worked out extensively in tomato, and we have our own BT brinjal, which is the only transgenic plant or genetically modified plant that has been commercialized in India, because for some reason, which is definitely the ethical reasons and the political reason, that none of the other transgenic plants has been commercialized, though many labs have been working and many are ready to be commercialized. For some reason it's not been, but BT brinjal is the example wherein they've tried to create a transgenic brinjal plant against the bollworms. Okay, and that is a very good example. Likewise, across the countries wherein the genetically modified organisms or the plants have been commercialized, in US and all, you have many of the plants which are herbicide resistant. You have plants like rice, which is drought resistance, or the tomato or any other capsicum plants which are tolerant or resistant to many of the pathogens. So it's been a routine mechanism. Either you target a particular gene which is known to have its role to fight against a pathogen, or can tolerate any stress. It might be saline stress, or it might be temperature, cold, or a very high temperature, or it might be water deficit conditions. So you have to first identify the genes, then try to work out. Okay, you can't create a magical solution overnight. Okay, you have to work out. So enough homework has to be done. Then you can actually target. And you also have a model plant in plants, especially the Arabidopsis thaliana, where it was sequenced 2000 and you take lessons from Arabidopsis thaliana. So that's a weed. But you need a crop plant to take a lesson, for which we have rice plant. So taking the targeting the genes in rice, we know that which gene is targeted for abiotic or against a pathogen or for drought resistance. So we try to pick up a gene in tomato or capsicum and try to work out and ask the question, like if I express this gene or knock out this gene, whether it can tolerate a particular stress or resist a particular pathogen. So that's how we work out. Or you can go for gene editing. Again, gene editing, you can randomly do it. And doing targeting for one guide RNA, you can't get a gene-edited plant against any abiotic or biotic stress. You have to generate many guide RNAs and try to work out in a particular plant to get ultimately a gene-edited plant wherein it will confer a resistance against a particular pathogen or tolerate any of the like water deficit or a saline stress. So one thing I tell you, whether you go for transgene technology or gene editing, enough optimization has to be done. Without optimization, you cannot directly target any gene, and at least you should have an idea like what that particular gene is doing in other plant where it is well like identified and well worked out. So we take either Arabidopsis or we take Nicotiana, that is tobacco, or we take rice plant and try to see. Okay, and don't ever come to conclusion that within overnight you get a plant. No, you have to work out whether you work it in a plant or an animal. Functional genomics is the same. You have to identify, work out, optimize, and then come to a particular product.
Thank you so much, ma'am. So now one last question for the day. So if all of you can just tell your views on how you are using it. So basically, what do you think is the role of artificial intelligence and machine learning in genetics? It would be great if you could just share if you are doing some kind of work related to the same. So first, I would like to go with Dr. Manjula now.
I'll come with some of the work what is being going on with artificial intelligence in the medical aspect, and it is very interesting to know how intelligence has been used in this part. But basically, currently I'm not working much on that. But yes, I look forward to work on it, and the aspects of how the treatment options can be worked with the patient, and the devices, how they have been managed with artificial intelligence, the drugs, the delivery systems that have been worked on. That is what I have come across with some of them. So I think this will be a very interesting part of work in future, probably. I think in just coming years, probably. So this will be very interesting to work on. So I look forward for working on the same aspects too.
Yes, ma'am. Thank you. Dr. Rajeshwari, ma'am.
Yeah, I know this is the era of AIML, and it's the buzzword. So any domain you see that we wanted to apply this AIML. So when it comes to the medical field, especially, of course, Dr. Manjula has mentioned, but to be very specific with regard to cancer, of course, the current research in this machine learning is for screening of cancer, especially when we talk about this, it's the image analysis. A lot of work is that which is being done, and we are also trying to initiate a little bit in this particular area by connecting with the computer science department with deep learning, then how we could do the study this pathological images, that is a pathology, and then which will help in the diagnosis, the prognosis. Of course, that is that which will lead to the personalized treatment and all. Dr. Manjula already mentioned about the drug discovery and all. So mainly we can use this the machine learning algorithms by which we could discover, even if there are very small, that is subtle associations are there between the genetic and molecular markers, because my concentration is on the identification of molecular markers, how this can be used for different types of cancer, which will eventually lead to improved classification, that is the diagnosis, and then finally into the treatment, because the identification part is that which is very much important. And more on this particular aspect is like which will help even the radiation oncologists where the planning time which it can be reduced. So like this, mainly the detection, diagnosis, and then the classification where you can optimize the cancer treatment and identify new therapeutic drugs, that is in drug discovery. I think these are the areas which people have started exploring using this machine learning algorithms. So maybe in future, yeah, if everything goes well with collaborations, this would be possible, and we would also get initiated. Thank you.
Thank you so much, ma'am, for sharing your insights on how AIML finds its role in healthcare with a focus on cancer. Now, moving on to Dr. Ashwani sir. So, we all love food, and I'm coming back to food again. But then, many of us actually wouldn't have thought of how AI and ML can find its place in the food industry. It's a question that I don't think many of us would have thought of. So, sir, can you please discuss if AI and ML has some role in the food industry specifically?
Interesting. The biggest problem is shelf life. And when I say shelf life, the packaging. And when I talk about packaging, the most prominent thing is what about how you have to build up an algorithm, how you are going to do the predictive modeling is particular component. And when I say this, the entire mechatronics concept, entire concept of packaging material, what is the food, what is the nature of the food, how it has to be packaged. Believe me, when we were children, we have only one thing that is called as soft tea. That exists today also. Nothing can beat even today. But now we have something called as composite material. Yes, ice creams are there, eat that pakra also. So when I say that, do understand, and as you said, yes, how genetic algorithm or AIML can be do it. Best example is this golden race pathway analysis. Entire, because pathway, if you talk about, it is a cobweb, and if you encroach into one point, you cannot conclude anywhere else. And this AIML is the one that can give a ray of hope. Yes, for us, it is easy which enzyme can work, which metabolite can work, how that feedback inhibition or feedback mechanism can enhance or upregulate or downregulate the particular metabolite, either for the taste, texture, aroma of the food, or the shelf life, or with respect to entire procedures. And when I talk about this, the most important comes in all the regulatory affairs. Today, all the regulatory affairs which are there, they have a subset of AIML into it. Whether now I don't say about, yes, there's something called as FDA, now we don't follow any concept of FDA, we have something called as FSSAI, which is a conglomeration or the blended version of FDA, thanks to Indian government. And in that, there is a very major share of AIML. So yes, in food, AIML was there and it will remain there.
Thank you so much, sir. Dr. Nagesha ma'am, would you also like to add on how AIML is playing a role in genetics?
Okay. It will be very generic, like I'm not targeting plants, animals, or microbes. Definitely, if you want to work on, I don't want to speak much about classical genetics because if I speak on classical genetics, definitely it will be more boring and it's too complicated. So let me try to speak on the molecular genetics part. What I like to say is, earlier we used to work for years together to identify a particular gene. Now the data sets are so huge, you can take it and try to, through AIML, like earlier it was the omics technologies that has ignited our minds, and now it is AIML which has lit the ignited minds. Okay, so now we have to come out with omics, which is attached as a suffix, like AIML omics, definitely whenever we are talking about this. And we can, with the huge database that is already available, we can target a particular gene in any plants, okay, whether it's a medicinal plant or a crop plant, and try to put forth the hypothesis and work on it. And definitely, like if I'm working on any of the microbes, if I add this particular metabolite into a media, a compound into a media, probably I might get a prediction stating that it might enhance a particular product. Earlier, it used to take years together to test it. Now, because of the already available data, we can actually cut short the time and try to work out. And as Dr. Ravi Shankar has already mentioned in marker-assisted technologies, which he has been working on and which India largely depends, because we largely don't depend on the transgenic plants, we depend on the molecular breeding, which is aided by the marker-assisted technologies. To identify QTLs, it's pretty difficult. I think through AIML, now you can easily predict the QTL, and we have a QTLomics. So definitely AIML will largely aid in cut short or predicting the work, and definitely will be a value addition for genetics. Thank you.
Thank you so much, ma'am. So we had asked one more question to Dr. Savitri Bhatt, ma'am. The question was, how does in vitro regeneration of ashwagandha plant contribute to therapeutic compound development, particularly with genetic modifications? And ma'am's response. Certain physical factors such as light, its intensity, humidity, temperature, as well as genetic makeup, that's the genotype of the plant, can cause variations during in vitro regeneration process. These variations, also known as somaclonal variations, have known to increase the production of vitaferrin, a steroidal glycovithanolide lactone. It's a type of vithanolide which is anti-cancerous, anti-inflammatory, and that was initially reported to inhibit cell growth and induce apoptosis in vitro and in vivo models of breast cancer. It also shows that it activates p53, disrupts beta-catenin, tubulin, actin elements. So WA has been proven to have potential to be a therapeutic agent for neurodegenerative diseases such as Alzheimer's. Thank you so much, ma'am, for sharing your response. With this, I would like to conclude the panel discussion on genetics. It was my pleasure being the moderator and hosting all of you for this session. Thank you so much for your invaluable time and insights. Now I request Dr. Vidya Niranjan, ma'am, HOD of Department of Biotechnology, Ivy College of Engineering, to address all of you.
I know this is the lunch time, just 2 minutes. Thank you all. Thank you, Dr. Ravi Shankar, who is my collaborator, who made his time to come for this panel discussion. And also, thanks Radheshwari Madam as well as Manjula and my own colleagues Nagashri and Ashwani. I think without them, our department is definitely will have there is no vacuum in genetics. So they are the pioneer in our department on genetics. So I hope all the students please take their expertise also. And thanks all of you for accepting information technology to be a part of genetics. That is what we are looking out for. Thank you so much for all the things, and also accommodating the pre-port. I have told in my initial talk why it was pre-port. Thank you so much for accommodating. And we will have like this many more in RVC. Okay, thank you so much. We will break for lunch, and then this is the end of today's second day pre-conference. So we will meet tomorrow at 10:30 for other. I hope students enjoyed, and also all the people who are watching YouTube. I think we have given invitation more than 600. So thank you so much for all of you, your valuable time, and I hope you have taken something back from today's panel discussion. Thank you.
Thank you, Manjula ma'am and Rajeshwari ma'am, for joining online. I request Vidya ma'am to...
Yeah, thank you very much. Thanks for the invitation.
Yeah, thank you very much, ma'am, for the invitation. Thank you so much.
Yeah, I request Vidya ma'am to honor Ashwani sir and Nageshwari ma'am and give away the samples.
I also want to tell that they have back to the Best Researchers Award for Teachers.
Thank you. Thank you. Thank you.
Thanks all. Yeah, we'll disperse and please come back tomorrow at 10. I know you all must be hungry. Your Sumatra told your students are being over now.
Thank you, everybody.
It is possible and it can be the right position.
See you all tomorrow at 10:13. Chemical.